Bonjour à tous. Bonjour à tous, bienvenue à cet exposé. J'ai le grand honneur de vous présenter Leonardo Trujillo, qui est professeur à l'Institut Tecnológico de Tijuana. Il travaille dans le département d'électronique et d'électrique engineering et l'Engineering Science Graduate Programme. Alors, Leonardo Trujillo a reçu un nombreux prix, mais il a aussi reçu de très beaux diplômes. Donc, il a reçu un diplôme d'électronique engineering, un master en computer science à l'Institut Tecnológico de Tijuana, et il a effectué un doctorat au CICESE Research Center dans la ville d'Ensenada, qui est aussi une ville mexicaine. Il est impliqué dans un grand nombre de recherches interdisciplinaires dans le domaine de l'évolution artificielle, de la computer vision, du machine learning et du pattern recognition. Plus particulièrement, ses recherches se focalisent sur la programmation génétique et donc il développe des nouveaux apprentissages et des nouvelles stratégies de recherche basées sur ces paradigmes. Alors, il a été le leader de plusieurs programmes nationaux et internationaux de recherche. Il a reçu plusieurs distinctions du programme scientifique mexicain. Ses travaux ont été publiés dans plus de 60 revues internationales, 60 conférences. Il a écrit 18 chapitres de livres. Et il a édité aussi 4 livres dans le domaine de l'Evolutionary Computation et de programmation génétique. Il est dans le board éditorial de plusieurs revues internationales, GPEM chez Springer, MCS chez MDPI. Il est reviewer pour des journaux de très haut niveau. Il est aussi organisateur de nombreuses tracks dans des conférences telles que GECCO, CECE, EuroGP, PPSN, CVPR et ECCV. Donc c'est vraiment un très très grand honneur de l'avoir avec nous aujourd'hui. Leonardo, thank you very much for being here today. Thank you.
Thank you, Perrick, for the introduction, and thank you for the invitation to the talk. I will begin by apologizing for my broken English, that I'll try and be as best as possible, and for my non-existent French, which even though I know Perrick now for many years and I've been to France many times, my French is not is not something to use in public. So thank you for being here for my talk. So I will begin with, well, here's the title of my talk, which is Dealing with Outliers and Multiple Sources Using Ransack and Genetic Programming. So there's a lot of things there mixed together in this title, and I will try and explain each of them in turn. First, a little bit from me, where I come from. For those of you who do not know, Tijuana is right there at the upper left corner of the Mexican map, right on the border with San Diego in the United States. Maybe you've heard of my city from popular songs such as the ones by Manu Chao. And here's some pictures of the city for you, for those of you who have never seen it. As I said, we are a border town. I would say it's a very special case of a border town between two countries. So very interesting to get to know the city. If anybody of you wants to come, we welcome you with open arms. Just some notes for those of you who might be interested. Tijuana is one of the leading cities now around the world in the culinary arts, and also in the beer production. Here's a particular brewery I like. And also, there's a lot of things nice here in Tijuana, as well as my institution, which we recently, in fact, tomorrow is the celebration of the 50-year anniversary, which I know is kind of young relative to some of the European institutions, but for us, it's a big date. So Pierrick already told you of our work. I'll just highlight that for years I've collaborated with Pierrick and with Bordeaux University in several different projects, and it's been a great pleasure. No? In fact, I visit Bordeaux a few times. Here I am with my wife. Here I am at La Victoire, the Church of Saint Emilion, and INRIA in Bordeaux. So it's been a great pleasure to get to know your city before, and I wish I was there in person for this event, but I've seen it before. And of course, thanks again to Pierrick for me getting to know your city and your institution. Would not be possible without his friendship and his collaborations. So the goals of this talk are fourfold. I want to introduce evolutionary algorithms and genetic programming to you, assuming that most of you maybe are not familiar with this. Explain to you how genetic programming can be used in engineering and IoT, and go to the main topic of symbolic regression and how we can deal with outliers and other forms of data corruption, in particular multiple sources, using random sampling techniques. No? So just a very brief introduction to what evolutionary computation is, and we will take the high-level definition given by the work of Darwin, no? Which is basically the context that is used today. So evolutionary system is defined by three elements. The first is an ability to store and copy packets of information. This allows for descent. A second is a mechanism to allow for variation of existing information, what is normally called random mutation. And a form of differential reproduction, which is normally understood as natural selection. So as it says in the first point, we want to have the ability to store and copy packets of information, but these copies will not be perfect. They will have random variations, and random variation is key in the process of evolutionary system, and also is key in the techniques I will describe later on. And where can you get to using such an evolutionary system? Well, here is we of course are all familiar with biological life. No, here's a nice picture of the tree of life as defined by biologists. No, and we can see that all of the designs in life are basically product of such an evolutionary process. I will skip the small video and just comment on what it presents. This is the Microbial Evolution and Growth Arena, or MEGA, from Harvard Medical School and the Technical Institute of Israel. And it shows here a real-life example of evolution in progress. In fact, I'll put play. I think we will have enough time. And here you will see bacteria in a Petri dish, and in the ends of the Petri dish there is no antibiotic. And as you go deeper into the middle of the Petri dish, the concentration of antibiotic increases. Now, this bacteria is fluorescent, so we can see it under light. And we can see that as the bacteria moves through the Petri dish, it will start to reach the boundaries of the Petri dish of different concentrations of antibiotic. And you will see that it cannot pass these boundaries until a variant that is resistant to the antibiotic can do it. So we can see almost in real time, this is a 7-day or 9-day experiment, as evolution progresses in front of our eyes. No? And each time we hit a boundary and the bacteria cannot cross it, you'll have to wait until a random mutation allows for the descendant of the bacteria to move along the Petri dish to continue to propagate. I love this example because it clearly shows how natural evolution works, and it also gives us a glimpse of one of the branches of what I just showed before in the previous slide, which is the tree of life. No, so we can see it here extending. For those of you wanting to know more, here are 4 very good books that I would recommend. The first one, of course, by Darwin himself. The others by 2 prominent presenters of the evolutionary theory, Richard Dawkins, and the philosopher Daniel Dennett, and a book dealing with randomness in evolution and its importance by Curtis Johnson called Darwin's Dice. No? So for those of you interested, here are some nice reading materials. So important takeaways from what I've just talked about in computer science. Well, Darwin presented natural selection by highlighting how artificial selection functions if you read the Origins. And in this sense, artificial evolution has been with us for quite some time now, not only before even computer science was here. Another important takeaway is the gene-centered view of evolution, that specific traits are what are passed on over generations and what are the focus of the evolutionary process. Another is the view of biology as engineering. So the process of evolution shows us a way of how to solve engineering problems, which is different in how we solve them as humans, though. Biological engineering is bottom-up, it's parallel, it's asynchronous, and in a sense, purposeless. So it gives us a different way of attacking engineering problems. And as I mentioned, the importance of randomness in evolutionary design, which is variations are blind with respect to function or to fitness, or in other words, are blind to the path that evolution is taking. And this is an important aspect in how evolution happens in biology and is leveraged in our algorithms in evolutionary computation. So here's a basic example. No? So say you want to design an antenna. How do you use an evolutionary process where you define the solution representation, our information packets to represent an antenna, say for example a vector, an array, a graph. Then you define a variation mechanism that will produce new antenna designs based on this representation from previous one with random modifications, and define a fitness function. We need to measure how good the antenna is for a specific problem that we want to solve. And the loop is basically this one. No? So we start with a random population or set of these computer representations of our antennas. We decode them and obtain the antennas in the real world or in a simulation usually, and use a fitness function to measure the performance that each antenna gives. So these second two blocks happen in the problem domain, and the rest is happening in the search domain of our task. No? So we do selection based on fitness. That is, we prefer those solutions that are better fitted to solve the task that we are interested in. We perform the random variation that we defined previously, and we iterate this process over the evolutionary cycle, with each iteration defined as a generation of the search. So this might seem a trivial process in general, but and you might ask yourself, is it really possible to design an antenna in this way? And I chose this example because I one of my favorite real-world examples of genetic programming, which is now quite old but still impressive, which was an antenna designed for NASA Space Technology 5 mission to measure the magnetosphere of the planet, and basically represents the first evolved design to go into space. Interesting is to see the type of design that would have been done with traditional engineering methods and the type of antenna produced by this evolutionary process. What I can say is that the evolved antennas did outperform the man-made antennas in all relevant performance metrics and were used by NASA to go into space. There are many forms of evolutionary algorithms. Black box optimization is probably the main application area of this type of algorithms, where you would find genetic algorithms, evolutionary strategies, evolutionary distribution algorithms, and others. And we can also use, as I showed in the previous example, evolution to solve design and learning problems, which is where genetic programming comes into play, and where we want to evolve variable site structures that may perform some form of computation, be it digital or analog. For those of you who wanted to know more of genetic programming, well, here's the the first book on the subject by John Cosa, which is regarded as the inventor of the technique, and two other important introductory texts for those of you who are interested by Langdon, Pauly, and McPhee and others. So how is such an algorithm used in other fields? Where I showed you the antenna design, as I told you, John Cosa, regarded as the inventor of GP. At one point, the popular science was said to have an invention machine. John Cosa used to run his evolutionary search and produce designs and then patent those designs. And he has many designs produced in this fashion, be in different domains, such as lenses, circuits, controllers, and other domains. Another nice example is the programming of quantum computers. This is a work by Lee Spector, where he produced the novel quantum computing algorithms using the search process. A machine learning tool that is very popular is called EuroGP, which is now part of DataRobot machine learning libraries, and it uses a variant of genetic programming to produce interpretable models. And we'll come back to this later on. Another nice example is TIPOC, which is an auto machine learning algorithm. So basically there, evolution and genetic programming is used to derive complete machine learning pipelines with little or no human intervention in the process. For example, it was being used by the World Bank. And Google is now using such techniques, including genetic programming and other evolutionary algorithms, to design complex deep neural networks. I'll show you two quick examples of our own work. For example, here is our work in collaboration with Pierre and also our colleagues in Tijuana from the CICESE Institute of Technology, Jose Enriquez Beltran and Victor Diaz Ramirez. This was part of the master's and PhD work by Enrique and under the supervision of Victor. And they collaborated with us on designing, I'll show you quickly the video. So the problem is haze removal from images. So say you have a scene, and the scene is corrupted by air light and other diffuse particles, which define haze in the air, and they make the image blurry and fuzzy. So the goal is to remove the haze from the image, and for this you can use standard approaches that model the haze in the scene and the image formation process. What we did was, so the goal is to have a degraded image and from that extract a haze-free image. And here we evolved a filter to accomplish this task with genetic programming. Here I'll show you just the result of the hazy image on the left and the hazy video on the right. That's in simulation. Here is in a real video sequence. Again, the evolved design was done with genetic programming, not with standard approaches. And here it is working on real time. Victor and Enrique implemented the evolved algorithm on a GPU card, and we could do real time haze removal with the evolved technique. Another quick example of working with my student, Victor, and with Pierre as well, was for the automatic improvement of a software system. So here you start with a working software system, and you automatically improve some of its performance characteristics using evolutionary search. In particular, we focus on the SLAM problem, which is simultaneous localization and mapping. And basically here we have a camera that's moving in a closed environment, and we need to produce a 3D map of the surroundings and localize the camera or the robot in this environment. So we took a fully functioning system and tried to improve its non-functional properties, which was basically to avoid errors and to improve its efficiency. And what we got using the same technique I described before was can be seen as follows. On the left, we have two video sequences of the improved version of the SLAM system, and on the right, the original SLAM system. As you can see, both of them produce very similar 3D reconstructions, but the one on the left, both of the videos on the left have a much better frame rate and in fact avoid crashes, as the example on the top shows that the original version crashed on this test case. So again, this improvement was done with the evolutionary process I presented before, and basically this shows you can also apply it to already working systems that perform some form of complex computations. To finalize the examples, I've just described works by others of evolutionary algorithms and genetic programming in IoT, which is the focus of this school. So you will find a lot of works of people using evolutionary algorithms in optimization tasks, such as the recent work here by Ngui and colleagues, where they use evolutionary algorithms to optimize task scheduling. This is a very common form of application of evolutionary research, and you will find many in the relevant literature. As for GP, which is more in the design or machine learning aspect, one popular application of it is to develop soft sensors. So a soft sensor, or also called inferential sensor, is a sensor that measures a variable or parameter of the environment of interest, but does it indirectly. Maybe this parameter is difficult to measure with a real-life sensor or costly, and you could do it indirectly by way of other measurements. And for example, Gordon and Smith were the first to work on such an application when working at Dow Chemical, and they produced various soft sensors that were used by this company in the early 2000s. And recently, you can find other examples, such as this one from the Biochemical Engineering Journal. For an interesting paper, for those of you who want to see an interesting analysis of the application of genetic programming in IoT technology, here's a book chapter by Bill Wurzel, which is called The Evolution of Everything in Genetic Programming, where he tries to sketch out a way in which genetic programming can be integrated into IoT technologies and used to do large-scale evolution on site, and maybe produce designs and solutions that were not feasible as we do evolution currently, which is, of course, offline. No? So I highly recommend this paper for those of you interested in both areas. So I'll finalize this introduction of genetic programming with a talk on symbolic regression, which is basically a regression problem where the goal is to find the best model, but also to have an interpreter model. So we don't start with a predefined, for example, linear model and just do parameter fitting. We want the evolutionary process to find the model and also the best symbolic expression of this model without any previous definition. No? Here is, for example, a simple example produced by the Arica software I mentioned before, where you have the data points in blue and you have in red the model, but also the symbolic expression of the produced model for this data set. So basically the goal is to find the symbolic expression. In this case, the search space is the space of all possible symbolic models for explaining the data in our problem set. Here's a good video showing the process by University of Applied Science in Upper Austria by Gabriel Kronberg and colleagues, which I will use to illustrate this process. In gray, you have the data points with the crosses, and the full line in gray shows the best model produced so far. In orange, you see all of the models in the population as it evolves towards the optimal solution. And you can see how this particular run has a convergence of most of the population to similar models, and you have a good fitting of the data points as evolution finishes. And you also, here you cannot see it, okay, there it is. Below you have that symbolic expression that describes the model, the data that was given to the genetic programming learning process. So that's the goal here in symbolic regression, to produce the expression below that best fits the data in our learning task. So difficult scenarios for symbolic regression, which would be difficult for any learning process. Of course, noisy data, too much or too few data could complicate things. Discontinuous data are also need special attention. And of course, always complex linear relations makes modeling more difficult. Here, we will talk about two cases, which is outliers and unlabeled multiple targets. So an outlier is a measurement of a system that are anomalous with respect to the true, of course, unknown behavior of the system. That's one of the difficulties of dealing with outliers, is that we may not know the true behavior, but we can assume that some of the measurements are not in line with the true underlying behavior of the system of interest. So here we have what we consider inliers in blue and an outlier of this time series in red. So why do outliers occur? Or they can occur by gross error, missing data, misaligned data, and we can have of different types: univariate, multivariate, and time series. So there are many ways to handle classical approaches. Are robust regression techniques such as least median squares or least mean squares. I will not go into details of each. Another approach, for example, for time series is the Hampel identifier, where we detect an outlier based on the median of the window around our time series. And if we see a point that is far beyond the median, we can replace it so we get a new data point which is in line with the rest of the data. Of course, if the amount of outliers in a specific window is too high, we can do the opposite and corrupt the data even further. So this relates to the breakdown point of robust regression methods, which is the sensitivity of least square methods and others, such as the Hampel identifier, to outliers. And this is referred to the breakdown point, which is given as a percentage. So the breakdown point is reached when a certain percentage of outliers is present in the data set, which produces an arbitrarily large bias in the final model. The highest breakdown point for methods such as the ones I described before and others is 50%. That means if we have more than 50% of outliers, then the methods cannot guarantee to produce a good model for our data. So what did we do? So we started the effect of outliers in symbolic regression, and we started by taking a data set. Here is the energy heating data set. I can give you more details if you're interested, and corrupted it with many different amounts of outliers, say 10%, 50%. Here is an error, it should be 90% at the bottom. And we just evaluated the symbolic regression population on all of the data, okay? But we differentiated the performance on the inliers and the outliers. And we can see on the top of this plot, the lines indicating the performance of the data on the outliers. And there we can see that even evolution progresses, the models do not fit the outliers at any moment. The error maintains stable across generations and does not change. So we cannot learn with GP on the outliers as we are introducing them. However, on the inliers, we see a difference in performance. We can see the algorithm converging, even in the initial generations. This told us that even at the start of the search, just by random population of models, we have a difference in performance between the inliers and the outliers. So we use this to derive a outlier removal method using just the random population at the start of the search process. So we contaminate the data set with different percentage of outliers. There's a typo there, it should be outliers. We generate 20,000 random GP models, and we use each model to sort the data samples based on the residuals to see the difference in performance of each random model on the inliers and the outliers. And we rank the data samples and keep the p percentage of samples with the lowest residuals. We are trying to see if when we keep those with the lowest residuals, these will correspond to inliers in our data set. And of course, the performance was not perfect, but we did see an interesting result. We could see that and particularly in the last two cases on the right, these plots show the percentage of inliers found when we keep those with the highest amount of, with the lowest residual errors, and the fitness of the individual on the bottom axis, on the horizontal axis. And we can see that a large percentage of inliers are found on a specific group of individuals which are not of the highest fitness, but they are of not also of the worst fitness of the random population. But we can see these clusters forming in these scatter plots. And we can identify those clusters of individuals as those individuals that produce the most amount of inliers when we perform the filtering process we described of keeping the data samples with the lowest residual errors. So we can fit there a kernel density function and use this kernel density function to choose a subset of the symbolic regression models to extract which data samples are possible outliers. So basically, we contaminated the data set with outliers. We define a cutoff percentile to choose where is our cutoff to take the data samples from the data set as inliers, and we do the following process. So we generate a random set of programs. In this case, GP trees I did not mention, but the representation of the models are trees, but you could use others. We evaluate each model, computing their fitness value, and we estimate the kernel density function in the scatter plot I showed before. Then we select a subset of these trees and compute the average residual error of all the data samples on using this subset of trees. And then we sort based on the average residual error on all the programs, and then we cut off using our percentile defined as input, those data samples below the threshold. And in this way, we keep those data samples that we assume are inliers. And here are the results. So we show the second plot, the blue line here, which is the result of the algorithm I described before. And we can see that as, of course, the contamination gets worse, performance degrades relative to the percentage of inliers found. But still above 50% contamination, we still are able to recover above 80% of inliers here at 60%. And even at 80% contamination, we are still above the breakdown point of 50% of most standard methods. So with this simple heuristic of just randomly sampling models in the problem, using the problem data set and basically ranking the data samples based on the residual errors on the random set of models, we can identify with some good confidence which data samples might be outliers. I will also describe a different technique for dealing with outliers, which is, maybe some of you know this, a very old technique, which is called RANSAC, Random Sample Consensus. The algorithm is simple to understand. I will describe it using the slides by J.D. Matas. He did a tutorial at CVPR 10 years ago. And 10 years ago, it was RANSAC in 2011, 30 years into the future, or some sort of title like this. So it's now 40 years since RANSAC was initially proposed. So say you have some data samples and you want to fit a model. Here it is clear which are inliers and which are outliers. And basically you take a random sample of the data set and you fit a model using this random sample. And then you see the divergence of the rest of the data points to this model. And you keep those that are within a certain range of the model, which is called the consensus set of the model. If the consensus set is too small, then you discard the hypothesis of this model. You take another sample and another sample until you find a model that gives a sufficiently large consensus set of the data, and this is the model you keep. So RANSAC was originally published in 1981 by Fisher and Bowles, popular in computer vision algorithms, for example, part of what is called the gold standard algorithm by Hartley and Zisserman. Many variants have been proposed over 40 years. Here I'm only using the basic RANSAC algorithm, and it's used in other fields besides computer vision. Here are a couple of recent examples in bioengineering and signal processing. So how do we use it for symbolic regression? Well, as was described in this image below, only that the modeling technique is genetic programming, so we can have such a noisy or corrupted data set, but by using random sampling and genetic programming, we can extract the model in the data quite efficiently. Even in such case, in this case, we have 90% of data corruption by outliers, and we can extract the original model. We have done this on several benchmarks and real-world problems, and the performance is quite stable and good, even in some cases, such as this example, when outliers reach above 90%. And I present this algorithm not only because of its use in outlier detection, but on the final problem I wish to discuss. So I'll just go back to our definition of symbolic regression, now using this figure. On the left, we have a standard regression data set where we have the features in the column of the matrix X, and the output or desired output is the vector Y, and each row here is a data sample. And so what we want is to find the function that gives us, given the input features of data, can predict the correct output that we are interested in given the data. So it's just the problem I defined before in a graphical representation. Now, multi-target regression is an extension of this problem, where what you have is, instead of one output for each input vector of features, we can have multiple outputs. So here in this example, we have 3 possible outputs for each of the data. So in multi-target regression, we want a model or models that can model each of these outputs correctly. You can do this independently by splitting up the data and learning as if it was a simple regression problem, or you can do it holistically and try to solve all 3 modeling problems, for example, in this example, concurrently in a single evolutionary process or a single learning process. Now, we studied a variation of this problem, which was called unlabeled multi-target regression, where we have the same or a similar data set as the one we presented in the multi-target regression case, but our data set comes as the simple symbolic regression problem. That is, we have multiple outputs, possibly multiple outputs, but these are not labeled. We do not know if a particular output in our learning data set corresponds to a different, to which output it may correspond. And we also do not know how many of them are in the data set. So we could be just a simple regression problem, or we could have 2, 3, or maybe multiple outputs that we need to model. This is not known beforehand. So the data set we see is similar to a symbolic regression task, but we are assuming that in fact we have multiple targets all combined together in the data set, and we want to extract them. One way to see this problem, to imagine this problem, is a kind of a clustering task, where we want to cluster the data samples in our data set on the possible outputs that may be hidden in the data set. No? So how do we solve this task? Well, we do it with the algorithm I described before, which is the RANSAC algorithm. And we just modify it to consider this case where in the RANSAC, originally it was thought of as a way of eliminating inliers from outliers. So we can have 2 groups. Here we have n groups of the data, and we do not know how many of them are present. So here is just an outline of how the algorithm works, assuming we have two models to extract from our data set. We take, as RANSAC prescribes, we take a random sample of the data points. Here are those in red, and we model or find a model based on those data points. Here is the model produced, for example, with the red points taken in our minimal sample set. We define a threshold to see which of the data points are in line with this model and extract the consensus set. If the consensus set is too small, we discard the model and repeat the process until hopefully we have a random sample of data points that extract a sufficiently large subset of data points from our data set. And then we remove those data points, we keep the model, and we repeat the process. In this case, since now we only have one data sample in our data set, then the model produced will fit it, and we can extract all of the remaining points. And this is basically the modification of the RANSAC algorithm that we implement for the unlabeled multi-target regression problem. We call the algorithm multi-target RANSAC, and here is the pseudocode, but for time I will skip it. I think the images are quite clear. And here are examples of the results. So here is the same example there. On the left, we have the data set, unlabeled data set, and on the right, the data set with the labels assigned based on the multi-target RANSAC execution and the 2 models found in data. Here is a case with 3 models. On the left, we have the training set, unlabeled, and on the right, we have the produced models by the algorithm, which was the correct amount, 3. And we've gone up to 5 models combined in the same data set. Again, here are all of them. Here are the 5 extracted models from this final example, which was one of the most difficult cases we had. And in fact, in this case, we have some cases in which the algorithm does not perform perfectly. So we can have solutions such as this one, where we have an extra model extracted from the data set. No? So this is the type of failures we have sometimes, where we still extracted the 5 models in the data set, but also got a spurious model in the process. So with this, I can give you the main takeaways of the talk, which is which I hope you take home with you, which is the GP is a probable evolution-based learning method for automatic design and modeling. Symbolic regression is the main application of GP and used to generate human-readable models, symbolic models. Outliers and multiple sources of data can hinder the learning process in a symbolic regression task, and we can use random sampling in different forms, as I showed, to help overcome the difficult learning scenarios. I will finalize with a quick invitation to those of you interested. We have an open issue in the Mathematical and Computational Applications Journal. which is an open access journal, and where if you want, you can submit a work on numerical or evolutionary optimization. This is a special issue we currently have online for those of you interested in submitting a work to the journal. With this, I finalize my presentation. Again, thanks to the organizers, thanks to Pierrick for the invitation, and thanks for all everyone who saw this talk. Thank you.
Sorry, excuse me, I have to change my computer. Thank you very much, Leonardo, for this very nice talk. Again, it was very interesting. Please, the attendees, if you have some question, please, you can ask them in the chat. I'm not sure I'm in the right place to see the questions in the chat. Maybe François, you can check if it's here. Okay. Oh, sorry. Yeah, some questions are here, so I will start from the beginning. Okay. Hello. Okay. Okay, but it was like 10 minutes ago. We had this question from Pascal Doré. When you said that you generate 20,000 GP, what do you want to say, sir?
Yeah, I'm sorry if I was not clear there, but we generate 20,000 random GP models, which would be random. In this case, we're using a true representation for our chromosomes in the genetic programming system. So we would generate 20,000 random models expressed as GP trees. So that's what I intended to say there. I'm sorry for the confusion.
Okay, thank you, Leonardo. Another question. Yeah, it is a very good question. Is any tool to support this method?
We still don't have any tools publicly available. I mean, there's as a tool, we have the code repositories of my students, and I could share this code with anyone that's interested. But yes, we wish at a moment to have it integrated in an easy to use tool to be downloaded and just used on a data set. But not at the moment.
Maybe we can mention that some tools are available to do GP search. Okay, you have GP Lab. You have some tools on Python.
Yes. Okay. So if GP is what you want to do, there are many tools available. Some of them commercial. For example, I mentioned here Eureka because it's a popular commercial GP-based system. But there are also others. For example, GP Lab, written by Sara Silva, which is in MATLAB library. In Python, you have the deep library of evolutionary algorithms. And there you have a GP implementation. And there are many others. For example, there are some to run GPU cards. A recent one is TensorGP. And we are also developing a GPU implementation of some of our GP algorithms, which we hope will be publicly available soon.
Yeah, just to add a few words on this, Leonardo Trujillo presented some books on GP at the beginning of his talk. I could mention that one of these books is free, available online. It's a book from Bill Langdon. This book is free. You can download it. It's a very well-written book. So if you're interested with GP, you can read this book.
Yes, it's a Fuel Guide to Genetic Programming.
Yeah, this one. Yeah.
Yeah, which is now a little bit old. It's from 2008, but still an excellent introduction. Always. Yeah.
Yes, for the introduction, it's very nice book. I don't see other questions in the chat. I have some, but okay, I see the answers, but okay. There is one question which is always the difficult question. Here you manage a lot of outliers, and sometimes when we're going to conferences, when we give a talk, we have this question about, okay, you have more than 50% of outliers. So what do you think? Do you think that they are still considered as outliers? I just made a course on this a few hours ago, so maybe you could add something for the students if you have any additional comment on this.
This is a very good question, no? And we always get this question. I think it's more a reflection of the problem domain that maybe people are coming from and what they expect in a data set. No? So, for example, if you see the works in computer vision, where, for example, RANSAC was developed and is widely used, they start by assuming that the amount of outliers is above 50%. I mean, if it wasn't above 50%, then they will not be using such a technique as a random sampling. So I guess the problem domains allows you to visualize cases where such a task will exist, where most of your data is not useful for you to learn on. And of course, defining it as outliers or inliers may be a semantic thing, Okay, if it's above 50%, then don't call them outliers. You can choose to do this. I mean, I don't see a problem with this. But in the end, you want to separate the data, and you are assuming that some of the data, which is a minority, is the one that is useful for you. So if you use this idea and call it outliers or don't call it outliers, then it's just how you want to label it. But if the task is, I want to separate a small amount of data from this large data set that I know that is useful to me, then call it this, no? And maybe it's easier. But for me, I would call them outliers, but I would accept that if someone prefers to describe it differently.
I think it's the proper answer to this question, I think. Thank you very much, Leonardo. Do you have other questions, the attendees? No, I don't see other questions. I know that you use your tools to build the contaminated data. I'm not sure, but I think you didn't mention that you use also these techniques. Maybe you can add few words on this.
Yeah, so we use the sample identifier to invert the process, and instead of using it to remove it, we use it to contaminate it. This is the process. I mean, there are some methods for some problems where you could, in fact, I just saw a recent paper of, I don't have the name of the authors in mind, but where they build a tool based on PCA to control the amount of outliers they introduce into their data set. There, they were concerned with classification tasks. So it's a little bit different in the process. And I'm sure you could use other forms of data contamination to artificially add outliers. We have applied to a couple of real-world problems where we know there are outliers. One of them was prediction of stock market data. And we compared there with robust regression techniques, and we did achieve an improved performance on this data set. Now, we have not published this, but we have found that on some real data, we can improve performance. But then again, if you only assume there are outliers, but you don't have them labeled, it is always maybe tricky interpretation as to if the data set is just peak value or some discontinuous process being shown there, or if it's really an outlier in the data set. So yes, it's tricky to do it artificially as well.
Thank you, Leonardo. Thank you very much again for this talk. It was a real pleasure to have you today. I'm also very happy that your talk is a very good talk after my course. My course was like a small introduction on Outliers. So thank you very much for this. thank you again. I hope to be able to work with you soon on this topic or another one. And you're welcome in Bordeaux when you want.
Thank you, Pierre. Thank you again for the invitation. Of course, you personally, I would not given this talk without your support, and all of this work was done in collaboration with you. Thanks to the organizers, and thanks again to all the students and everyone in the public who saw this talk. I hope you find something useful in it for you. And again, I'm sorry that I'm not able to be there in Bordeaux with you. I always love to be there and to visit. So I'll say digital hello and hug to everyone. And thanks again for the invitation.
Thank you very much. Thank you. Bye-bye. Thank you to all the attendees. Bye.